The last day we're going to be spending on lesson 7.3, and as you know, on the last day, the quiz today is going to cover the entire lesson. So you've got to go back and watch days 1, 2, 3, 4, 5, and 6. Obviously, remember day 4 did not have a video. It was a lab report, so don't worry about that. Okay, so go back and watch all those videos, including this one, before you actually start the quiz. The very last topic we need to cover is endo and exocytosis. Okay, so for the first couple of days, we were talking all about diffusion and passive transport. Passive transport is a type of transport that requires no energy because it's small molecules moving from an area where they are at a high concentration to an area where they're at a low concentration. Yesterday, we talked about active transport, so I'm just going to show you that picture again. In active transport, we do require energy because we are moving particles or molecules from an area where they are at a low concentration to an area where they're at a high concentration, which is not somewhere that molecules naturally want to go. This is a forced process, which means that it requires ATP. Now, today we're going to talk about a different type of active transport. This is the type of active transport that takes place when we are moving large particles or liquids, okay? If I want to move a large molecule into a cell, a lot of the time I can't actually make a protein big enough to allow those large molecules to enter. I also can't just expect them to go straight through the cell membrane. That's also not possible. So what the cell has done to fix this problem is that it created vesicles. All right, so the transport of large molecules inside and outside the cells requires the formation of a vesicle. Vesicles are small portions of the membrane which surround materials and transport them to other parts of the cell. So as you can see here, as a material decides to exit the cell, a larger material, it is surrounded by a layer of cell membrane. This thing is called a vesicle. It goes to the cell membrane and it starts to fuse into the cell membrane. And as it fuses, the cell membrane basically adopts this part of the membrane and this part opens and the material is released. And then this is called exocytosis, meaning things are exiting the cell. That's why it's called exocytosis. Endocytosis is the exact opposite, but it's pretty much the exact same process. This is where larger materials are trying to enter the cells, okay? So you can see here, these materials come, they push the membrane in, and the membrane starts to form this little thing around them until the membrane basically forms a circle and pinches off and turns into a vesicle. Now that these materials are in a vesicle inside the cell, this vesicle can be transported anywhere in the cell that they're needed, right? So if you need them in the nucleus, they can go to the nucleus. If you need them in the mitochondria, they can go to the mitochondria. Wherever they need to go, this membrane or this vesicle can transport that material to where they need to be, okay? All right, so exocytosis is a process where things exit the cell, so you can see the arrows. It fuses with the membrane and then the materials leave. Endocytosis is a process where larger materials or liquids want to enter the cell. So they squeeze into the membrane, a vesicle forms around them, and there they are inside the cell, ready to be transported anywhere within the cell that they're needed. So I'm just going to show you this simulation real quick. All right, so now you guys on your own, you can play this audio if you want, but I'm not going to, just because I will explain it. But it is really good, so you should go on your own and actually watch this. All right, so first thing, what happens is, let's say that this is a cell called a macrophage. A macrophage's job is to basically go around your body, finding different invaders like bacteria and viruses. They surround them, they eat them up and break them down, and then they destroy them. So basically what it does is that it found this little green thing, which is the bacteria. So this is the plasma membrane. The bacteria, it comes very close to the bacteria and then it starts to engulf it. Basically, engulfing means that you are forming like a little circle around it until it closes in on it, right? Now it is inside the cell, and the cell can start to destroy it and break it down and do whatever it needs to. The same thing happens when I want liquids to enter my cell. See? The liquids approach, the cell membrane forms around them, and a vesicle is formed on the inside of the cell which contains this liquid. Then this liquid can be moved to wherever in the cell that it's needed. Just a quick thing, boom, vesicle forms. And another different way that it can happen is that they can all come into here, different materials, and then it can close and we can get another vesicle forming in this way. Okay? That's endocytosis, meaning that a whole bunch of different types of material can enter in this way. And then exocytosis, here's the vesicle, and it's going to start approaching the outside of the cell. As it approaches the outside of the cell, the membrane fuses here with the cell membrane, and as it begins to fuse, it opens and the materials can be expelled from the cell. See? Boom. That's exocytosis. So endocytosis is when things are entering the cell, and exocytosis is when things are exiting the cell, okay? But using a vesicle. All right, so you guys, now remember, Day 6 quiz includes material from the entire lesson, so go back and review all of that. Good luck.